We're talking with Zaven Khachaturian, who I've known for decades, and so has one of our participants, Sonia. Sonia wanted to say a quick hello before you give a talk today. And Sonia said that 40 years ago, you funded her first grant.
Yeah. Hi, Zaven. Sonia and Coley Israel here. You started me off on my aging and sleep and Alzheimer's research, and now here I am as a caregiver of my husband who has PA. So different hats, but it's wonderful to see you again.
You were in San Diego, right?
That's right, yeah.
You were in San Diego at the time?
I was, and I still am.
Yeah. Let's see, I forgot the name of the guy that was a big guru in sleep research.
It was Dan Kripke. I was working with Dan Kripke back then.
Yeah.
And then I became the guru.
Good to see you.
Nice to see you.
Yes. Sonia, thank you so much. And hopefully you'll have some time you'll be around for questions. Sonia has to leave a little early. So I want to introduce Zaven Khachaturian. Zaven used to head research for all of National Institute on Aging, which is part of the National Institutes on Health. And he'll tell you a little bit about his background, but he has spent his whole career studying Alzheimer's and giving money out. And now he does consulting for the Alzheimer's Association. Zaven, you want to tell us a little bit about what you're doing with the Alzheimer's Association?
I'm a senior advisor to the Alzheimer's Association. I've done that since, really been associated with them since their founding. In 1995, when I left NIA, I became their senior science advisor, and my contribution was to revise their scientific review process, develop the new review system. Then I developed the Nancy and Ronald Reagan Research Institute for them. From that evolved the large conference that they have now, the AIC. Also, one of the proposals was to create a journal, scientific journal, which they did, and I'm now advise them on new initiatives, new ideas for scientific directions, do a lot of fundraising for them. I also work with universities, foundations, other governmental entities, primarily doing strategic planning. I worked with the Organization for Economic Cooperation and developed for them an analytical report which led to the G8 Summit on dementia. I worked on the development of strategies for the national program on Alzheimer's disease, the one that's now increasing the funding for research. So those are the kinds of things that I do. Periodically, I write editorials looking at what are the reasons for lack of success in clinical trials, looking for new directions for research. That should give you an idea.
Thank you. And so let's talk about PCA, because PCA came on the scene in 1988. Before that, Frank Benson at UCLA, Frank found these series of patients who shared, they were very early onset, and they shared a visual and spatial phenotype. Do you remember that?
Right. I remember Frank. Frank was a good friend of mine. He was an outstanding behavioral neurologist and had some real insights about clinical entity. His unique characteristic was he was very close to patients and was an acute observer of patients' behavior. He not only was good, he also mentored people of the same color, people like Jeff Cummings and Bruce Miller, others who also become like him, astute observers of the phenomena and describing it very carefully. So he made some very important contributions, and I think his observations about the posterior cortical atrophy is a good one, made a very important contribution to the field, making the distinction between that entity and Alzheimer's.
Yes, and also so you've probably been around to witness primary progressive aphasia and other rare dementias. When you were starting out, I mean, did people even know Alzheimer's was so common?
That's true. Alzheimer's, the phenomena of Alzheimer's and all the variations of it, the different forms of dementia, have always been part of the human condition. And early on, people described it as rare conditions and attributed to aging. That was the scientific study began in the 60s in UK, when a group of psychiatrists, neurologists started paying more attention to the disease. Sir Martin Roth especially was important, and that set the modern era of studying the condition, that is the clinical condition, along with the underlying biology. When the NIA came on the scene in 1977, the first director was a psychiatrist by the name of Robert Butler, who had been a great advocate for aging, and he had seen the dementia and made dementia, Alzheimer's, one of the institute priorities. I was recruited about that time to basically develop a national program on brain aging and Alzheimer's. And the primary task at the time was to overcome the general thinking in the field. That was true in the scientific community, in the medical community, the general population, that Alzheimer's, the condition, was due to aging. It was synonymous and used to be called senility. So the point was what you can do. NIA took the position that aging per se does not cause the disease, and that aging, one can age quite healthy and function quite well, and that if a disease occurs, it's due to some underlying pathology which requires studying. So that became the major thrust and struggle at NIA. The other struggle at the time was to overcome the lack of interest in the scientific community in the problem of aging and the problem of dementia. The same as in the general population, the thinking was that there's not much of a future for a scientist to be involved in Alzheimer's. Some of the other barriers were, besides the scientific and the cultural issues, was the lack of money. There was very little interest in Congress to fund this kind of research. So those early years, the efforts were, one, to change the public awareness, make them aware that this is a disease and not a natural phenomenon of aging. That took a lot of work to change the public attitude. That required writing editorials, writing articles, and people like you, science writers, played an important role in getting the message out in the media that we have a condition here, a serious condition that's going to get worse, but that we need to study it using the modern scientific study of brain, neuroscience, psychiatry, neurology, bring them to bear on this problem. Along with that effort, my efforts took get the word to the scientific community and get them interested in the problem. But at the same time, trying to present to Congress a cohesive story that this was something worthwhile, that if we made an effort, we could turn this situation around. So those were some of the early struggles. We did not focus too much about the niceties of distinguishing the different forms of dementia. It was one big amorphous entity. In fact, terms like senile dementia of Alzheimer's type, Alzheimer's were used synonymously. There was the thinking that there were two different kinds, the early onset and then later onset. So people were arguing about those findings on changing that lexicon by getting rid of the term senile dementia of Alzheimer's and focusing on just the idea of Alzheimer's. So that was a major achievement. Then the next phase of development came with the idea of trying to develop criteria for diagnosis. So that took place in 1984. First, there was an effort to make a clinical diagnosis. Clinical criteria was created. Then I put together a workshop on the diagnosis, and we developed the neuropathological criteria. And since then, gradually, by creating the infrastructure to study the clinical entity longitudinally, we followed up with pathological studies. That was done through the Centers program. We began to sort out the fine distinctions between the different forms of dementia and define clinical features. So that was the general effort, and it's in a reiterative way, we're making progress in defining the disease. By 1988, people like Frank Benson came along and said, well, there are these fine distinctions that are different than the traditional Alzheimer's. Then the distinction between vascular dementia and Alzheimer's became apparent. Lewy body dementia became clear. Now we're making the distinction with tau PET. We're able now to dissect it in much finer detail, the clinical features. Anyway, that's where we are now.
It's interesting. And what we're finding out in terms of research is that a lot of patients with PCA are not even diagnosed, and they're misdiagnosed for many, many reasons, because they're not enough clinicians really understand it and know about it. But also in the research arena, a lot of patients, because they don't have the memory problems, they can't be included in the studies because the models used to test them are based on memory. You know, all the tests that they use to see whether anything works or not. Have you found that in your world?
You're correct that those fine distinctions are not made, and it takes effort to get the scientific community to focus on those fine distinctions. Scientists tend to, once they develop an understanding of a phenomena, they tend to put all their energy exclusively into that previous description, because that's what's going to get their next grant. They do not have the time or the interest to ask questions and look for the fine distinction. That's one phenomena that's going on, and it's been part of scientific effort to continue in a direction. It's like a law in physics. Once a mass starts developing momentum, it keeps going in a given direction. It's very hard for them to shift direction. It requires some tremendous effort for them to shift direction. Right now, the big problem is they're focused on two of the lesions of the disease, the amyloid protein and the tau protein, and those looking for understanding how those lesions are made, what the genetic basis is, what the chemistry is, consumes a lot of the effort. despite the fact that the clinical and research evidence shows that not every dementia patient has these features, and that there are distinctions. Those just do not get the attention. The other is the clinical community in practice, whether they be neurologists, geriatricians, or psychiatrists, they also follow the scientific community's lead and lop on to these ideas which become scientific orthodoxy. So they don't want to spend the time studying the fine details of the patients the same way that Frank Benson did. Frank Benson is really good anchor point to make that comparison. Here's a guy that spent time with the patients and studied them as they're exhibiting, expressing their symptoms, and started making some generalizations that these folks are different, that they behave differently, maybe the underlying pathology is different, that we need to treat them differently. That tendency is not there, and it's not going to be there in the scientific community because it doesn't make the literature or the package inserts in pharmaceutical companies provide, or the continuing education program to physicians do not provide that kind of detail. It's for groups like you, the attention of the medical community. In other words, educate them about the fine distinction.
What's so interesting to me is that, you know, you started off talking about early Alzheimer's, and we made this, you know, before, you know, it was 1987 before we had the first APP gene mutation, and it represented a very small number of people. Now we have a couple other genes that cause early Alzheimer's. But with something like PCA and primary progressive aphasia, which is a language dementia that most of you have heard about from me, this also occurs early, but no one knows why. There are no genes that have been really identified for these conditions. What are your thoughts?
Okay. Well, let's look at the big picture. Really, this group of disorders are what I call complex neurological disorders. They're not the traditional neurological disorders where there is one-to-one relationship between an infectious agent or a bug or a pathology that leads to a clinical expression of the disease in a linear fashion. These are complex in the sense that they have many moving parts. That concept of complexity of these disorders has not really saturated into the field. So that's one main block for progress in the field, for not dealing with complexity of the disease. I'll explain a bit further what I mean by that. The second important feature that we've learned is that these disorders start very early on, 10, 20, 30 years before the clinical features express. In all cases, they all affect one fundamental problem. That is the functioning of a neuron. Neuron is the simplest unit that makes up the complexity. You build up the complexity by connecting neurons into networks. And there are different networks that have one neural network that controls movement, another network that controls attention, another one that controls affect or emotion, another one that controls memory, vision, sensory process. Each one of these has within each of the networks there are fine details. When you look at attention, it's made up of different parts of attention. Selective attention versus general arousal that controls sleep part. When you look at attention, the cognition is made up of verbal skills, executive decision. So there are finer details. When these disorders occur, what's happening is that the systems are failing. In the case of Parkinson, the failure is primarily due to the network that's controlling the motor movement, whether the motor movement be the large muscle movement or the fine details. They also involve other parts of the brain that control affect. That's why dementia, Parkinson have affect with depression. They also have memory. So there's a lot of overlap in their functioning. And depending on the pattern of overlap, you get the different clinical features. Something like printing colors, doing color photography, where you have 3 primary colors and you can mix them and create all kinds of different hues. These neurological disorders with the different clinical expression are really like a color photograph, where the fundamental what's going on in the brain are these failure of these networks. And as I said, most of this process starts many years before. The complexity comes into the picture by the fact that no single etiologic factor accounts for it. That is, it's not a single gene, and it's not a single life experience, and it's not single loss of a particular neurotransmitter system, but it's a combination of them. And it may occur over the course of a lifetime. For example, it might start with changes in the vascular system that affects the metabolic activity of the brain in different parts of the brain. If you deprive the brain of glucose and oxygen, you set the stage for all kinds of syndromes. Depending on what part of the brain that's being affected by this, you're going to get the expression of different kinds of processes. So to understand the disease, you need to be looking at it from this perspective rather than looking for a quick solution with a simple etiologic factor, a single gene or a single molecule that's defective, and correct it. That approach, unfortunately, hasn't worked. It's worked in Parkinson with the dopamine because in that case, the relationship between dopaminergic system and movement was straightforward. In cognition, it's a lot more complex. We thought the cholinergic system was involved. Now we're finding out that, well, it's a lot more than the cholinergic system, and that the drugs that are available work for 6 months or a year, 2 years. After a while, they fail to be effective, not because the drug has changed, it's because the system that's going to benefit from it has been destroyed. So the name of the game is to find out what are the reasons why cells are dying and becoming dysfunctional in distinct parts of the brain. Clearly, genes play a role, but genes do not play a role by themselves. We are beginning to find out that many genes interact in creating these complex patterns. So in many respects, whether you're dealing with Alzheimer's or Parkinson's or any of the other forms of dementia, they have a lot of common features, but they also have very distinct differences. The distinct differences come into picture because of the manifestation of the sets of behavioral problems that the family caregivers have to deal with. So we have to separate how do you deal with the care issues, the agitation with the sleep loss, the hallucination, memory loss, define distinctions between them, provide the caregivers the tools, the knowledge they need in how to cope with it, how to have the primary care physicians learn to detect these very early signs so that you could start preparing the family members what's to come about. But at the same time, have the scientific community step back and look for more complex solutions to the problem. The simple approaches we're taking now with the drug development have failed. We've had 40 years of very exciting science, but very little product. Only really 2 classes of treatments have been developed. One deals with the cholinergic idea, that is the cholinesterase inhibitors. The other one is nimodipine, memantine, which regulates the calcium levels within a neuron. Besides that, we don't have anything else, but we need to be looking for other avenues for developing this treatment based on the notion of complexity of the disease.
Yes, and now they're like I'm writing a lot at Neurology Today on the link between these neurodegenerative diseases and the immune system. And if you look at Parkinson's, you know, with all the gut problems they've had, you know, and now people are just making those connections. Just now, it's very interesting. What do you think?
That's a very good, yeah. That's a very good point you're making. It's becoming apparent that inflammation might be playing an important role in this degenerative process, that early inflammation could come about because of breakdown in the blood-brain barrier, and that's why maybe head trauma or early minor vascular changes in the brain could cause leaky, break down the blood-brain barrier so that infectious agents can trigger the inflammatory processes. It's the inflammatory process that are ultimately coming about to kill the cells.
It's interesting. So what do you think right now in this, in everything you're looking at, where do you think the field should be going? You know, amyloid drugs, you know, now we have tau drugs that are coming to fruition. Where do you think we're going with all these medications?
Well, I think all of the observations we have, whether it be the tremendous knowledge we've developed about the amyloid, including the genes that are involved in it, how this abnormal protein is produced, how it's processed, the same thing with tau. All the different ideas, and there are lots of major ideas, are part of the story. I'm not questioning, no one questioning the validity of these observations. The question is, no single one can lead to the treatment because the disease is complex. So what we're trying to do now is put all of this together into a complex, complete map to look at the relationships of these various ideas. Which one comes first? What is the connection? Which one leads? Which one follows? And try to develop treatments that would be a bit more complex. For example, we know that circulation to the brain, availability of glucose is tremendously important to the viability of neuron. If you deprive a neuron, a nerve cell, from oxygen and glucose, within a short period, that cell will die. The model for that is stroke. In stroke, you have a perfusion problem that would destroy parts of the brain that's not being properly perfused within a few hours. Now, consider the same process where you're depriving the neuron of glucose over a longer period. But instead of having massive disruption, you just create a leaky situation where the deprivation is over a longer period. That model.
Just a second, Zaven. Let me just turn this off. And
All right. Are we back on?
yes, go ahead. Yeah, can you hear me?
That sort of gives you a picture of how something that starts way upstream, years before, that's going on undetected, slow problem with perfusion of certain parts of the brain where circulation is not 100%. That would selectively affect the cholinergic system because the synthesis of acetylcholine is very dependent on glucose. So now you have a relationship between the observation of cholinergic deficit with glucose metabolic disorder. Now, if you're going to develop a new treatment, you should deal with the early vascular changes followed with the cholinesterase inhibitor. That's just a simple example where you would be combining different modalities of treatment rather than dealing with a single pill. Another one would be changing the diet or exercise, or some of these other non-pharmacological approaches are beginning to show beneficial effect. So that's the model we're trying to develop. Now, this makes it much harder for clinical trials for drug discovery, development. It creates a complexity that they're not equipped to handle, and they're reluctant to readily get into it until we demonstrate how this would be done. So we're at a transition point in the whole complex neurological disorders, shifting from the way we've been doing drug discovery development to the new model, which would require more complex approaches. We're beginning to find out that there are a large number of biomarkers that allow us to track the progression of the disease and be able to detect them very early. That's a very lively field. It's because if you can find out by blood test or by imaging some early changes that are predictive, would allow you to take corrective action. We're beginning to find out that there's a very good relationship between cerebrovascular disease and later on dementia. In fact, we have a meeting coming up in Berlin, the World Health Summit, where we would be proposing the idea that we can perhaps prevent dementia by treating stroke or multi-infarct, microvessel vessel hemorrhages early on, and that these could have a subsequent beneficial effect. So these kinds of ideas are there. They're beginning to germinate and hopefully will give us new insights.
You mentioned diet. What can patients and their caregivers do today? Is there an optimal way to eat to make things better in the brain?
I don't have a particular diet, but I think the general advice would be to follow some of the already well identified diets, such as the Mediterranean diet, that are healthy and they do not have as much fat or sugar. There is a good balance between fiber and protein. A lot of vegetables, I think, are very useful. So I do not have a particular diet, but I think generally, I personally find changing my diet has improved my cognitive functioning. I've seen the beneficial effects of that. There is a major study out of Finland called Finger Study, where they've followed longitudinally the Mediterranean type diet, exercise, and keeping the patients socially engaged in mental activities have to have really great beneficial effect. I urge your audience to read the Finger Study out of Finland. I think it's been published. Unfortunately, I don't have the reference at my fingertips.
I'll get it for people who want it. And let's talk about exercise, because that's really important. And that's something that a lot of people with PCA might find more difficult because we're having visual and spatial problems and they can't drive anymore. We have some people, Shirley, you go to the gym every couple times a week. I know that, and other people have been exercising. Could you talk about the effect, the benefits of exercise?
Well, I think exercise can be, doesn't have to be necessarily strenuous. It can be as simple as taking good long walks or doing, raising the heartbeat a little bit. It would improve the circulation to the brain. It gets back to the point I was making about circulation, perfusion of the brain being important. A good exercise not only affects the vasculature, but also puts out a lot of chemicals that makes you feel better.
Yeah, I think I'm a big proponent on exercise, and the studies that I've read really suggest that it is probably, you know, because there's no medicines. You know, you were talking about the acetylcholine receptor, and Aricept, by the way, is the drug that many of you maybe have put on at least temporarily. Maybe it worked, maybe it didn't. Then you might have been put on memantine. Maybe it works, maybe it didn't. And now there are a number of studies on inflammatory anti-inflammatory drugs that may ultimately, you know, or retweaking the immune system so that the immune system can fight off inflammation. So, and in terms of PCA patients, what is your sense of what people can do to get themselves into studies in their areas, since it is it's a rare
there are lists of trials published by NIH. I think that would be one source to identify. The other one would be to approach the neurology department or Alzheimer's center nearby to see if they have a trial going that they might participate. The Alzheimer's Association and various organizations have a trial match program, so that kind of effort is now underway. UCSF and there is various groups have developed this protocol where they're trying to recruit large number of individuals that are ready for clinical trials. So I think afterwards, maybe we can exchange some emails.
Yeah.
You can try to find some of those sites for you that you can share. Yeah,
Jamie,
you have a question. I
Zaven,
see a hand.
here's the problem with the clinical trials is that because of the visual component, the PCA patients get excluded. They can't do any of the drawing tasks that are part of almost every neuropsychological battery. And that has excluded almost all of them. So that's what makes it so difficult to get into any of the trials. It's as if, you know, when blind people are excluded as well for the same reason.
That's a good point. I didn't think about that. I think you're making a good point there. Perhaps the thing would be to develop a position paper or some statement that could be produced by your group to NIH to indicate this as a problem area, and have the groups that are doing clinical trials make this a special attention. There is the clinical trial consortium that I created at many years ago. It was originally located at San Diego, then more recently it moved to USC, Clinical Studies Consortium run by Paul Asen, who is now at USC. That group might be a good one to bring this problem to their attention, and to have people that would be sympathetic for your point of view, be the spokesperson, somebody like Jeff Cummings or Bruce Miller. I think any of those people would be a good spokesperson to write an editorial identifying this as a problem area. If I receive an editorial or perspective on it in my journal, I'd be glad to publish it, sort of publicize the problem, that there is a problem that certain class of patients are being excluded for the point that was made.
I've actually spoken with Brad Beauvais, Doug Glasgow, Jeff Cummings, a whole bunch of people, neurologists who specifically see patients with PCA, and we've talked about what is the best approach, whether we do a investigator-initiated grant to some of the companies, but I will talk to them, and Jamie, maybe you and I can do this together about an editorial.
I think that would be
I'd love that. George Perry. Yeah.
Those are good people. If you bring George Perry and Doc Glasgow is great. Jeff Cummings, I think they might put together a short perspective paper.
Yeah, George asked me to write something for his group on PCA. I'm going to do that, and I just created a map project where I'm asking people on our Facebook page to let me know where they live. And so I've created a map of like dots of where people are in the United States, so that we can begin to really use that as a social map as well for people who, like here, some of you, and I know this to be true, had never met somebody with PCA before Barbara. The other day, you know, Barbara's new and she was just newly diagnosed, and she had never met anybody with PCA. And most neurologists, if they've seen one or two patients in their career. So, and I think it's, you know, when I started covering ALS and other neurodegenerative diseases, and, you know, we thought, oh, it's such a rare disease, but now we know it's not. And then we thought, oh, the patients are locked in their bodies, but their brains, their minds are okay. Now we know that's not true at all. And so we're beginning to understand so much more about these rare dementias. And I think there's going to be like a curve, a learning curve for neurologists to start really identifying more people and trying to figure out, getting involved with research and trying to figure out why. What is it? Why does it come 10 years earlier than and why is it, and does it hit the visual system? Is it something about our eyes growing up? Is it, you know, do we all wear glasses? Do we all have bad vision? Is there something about that? Were we not coordinated? You know, what is it? What are the factors that go into this phenotype? I'm going to open it up to questions now for a few minutes. If you have a question, just raise your hand and I'll unmute you. And anybody? I can't see everybody all at once. So if you wanted to ask something, just take yourself off mute and ask. No questions. Well, I'm going to ask a question since I have Zaven. Do you see a question, Zaven?
Go
ahead.
I wanted to make the point that you have really a unique case to make for including your group in clinical trials, because you have some clinical features that are of interest to the field. For example, the sleep loss is an important phenomena, and it's now becoming apparent that changes in sleep might be early indicators of not just this overall dementia. So I think that would be just one example of an angle where a trial could be designed to detect early sleep changes, to follow longitudinally their effect, to follow treatment. So there's a large area of these special features of the disease that could be important to understand the underlying biology and also how to deal with it. Can have beneficial effect beyond your group to the overall dementia population. Depression is another area where it's important for all kinds of.
I was hoping someone would mention.
You know, it's funny.
We talked about exercise and diet, so sleep's got to be in there with it.
Thank you, Sonia.
Marilyn, do you have any questions for Dr. Khachaturian?
No questions, other than with Lou, who is older than most. Lou was diagnosed 7 years ago with a few symptoms before that, and he'll be 79 soon. I would just add two bits, that there's been no cardiovascular history for Lou. He's absolutely healthy, as we say, from the neck down. He did have a traumatic injury about 45 years ago. He was hit by a car while he was out of the country on a business trip. And, you know, we can't, in spite of the broken bones and everything else, we just can't help but wonder if that traumatic blow might have gotten something started. I will also add that Lou, who's probably at about stage 6 now, had a pretty rapid progression in the last 6 weeks. For the first time, a depression has surfaced, and a very deep depression, and anxiety surfaced and quickly turned into some panic. So we're working behaviorally with UCSD's wonderful senior behavioral health, and we're able to moderate everything without any additional medications. So I'm feeling very hopeful about the treatment, but it is a pretty quick onslaught. And I guess my question related to that is, can there be some comments about, at this stage of the disease progression, is there that rapid change? I seem to read that it's not uncommon, but I'd be interested in opinions about that.
I'm not aware of any specific research that has addressed that issue of the rate of progression change. I don't think it has been well studied, but it's a important point because it could give us some insights. There is a slight glimmer of a explanation provided by some of the early work that Dr. Alan Roses did with ApoE, where he showed that depending on the particular combination of ApoE people have, the age of onset could vary, but he did not really pursue very deeply into whether, besides the age of onset, where there was a change in the rate of progression. I think that remains to be understood. So I'm afraid I don't have a good answer to your question, but it's a good one.
So noted.
It is good. And Marilyn, you know, I'm sure that his neurologist has also. Okay, so in terms of Lou, maybe something else happened as a trigger 6 weeks ago. Maybe, you know, a virus. Maybe something that you know, he wasn't even, you know, that you couldn't see, but maybe on looking back on it now, maybe there's a pattern of some change. Just keep an eye out for anything that looks different, you know. Anybody else have it?
And down the other, interestingly, I noticed what I call a bundle of symptoms that I brought right away to the attention of our primary care physician. And let me quiet that. And he saw Lou and made a note of the situation. And then it was 5 days later when the first panic attack occurred. Yeah, I can't get enough air. And it turned out to be paradoxical vocal cord or vocal cord dysfunction symptom, without any physiological basis. So there was something brewing, but all blood tests were normal. Urinalysis was normal. There was nothing lab test-wise, except we did find 2 tiny blood clots. Interesting, huh?
Interesting. Well, yes.
The case you're mentioning is really a good insight. Is the fact that he had a head trauma early on, which could set the stage for what happened. The head trauma could have, I'm speculating, could have caused some minor damage to the blood-brain barrier and set the stage for the kind of inflammatory processes that I described. Mm-hmm. When he was younger, the ability to withstand the inflammatory processes would have been resilient, but as some other problem, metabolic problem that occurred more recently could have broken down that resilience so that it's just like a dam breaking down. The flood sort of overtake. That's the kind of process. But what those factors are, it's just a guess on my part, but it sounds that there's a reasonable explanation what started the process, that initial head trauma. There is lots of cases where head trauma, no matter how minor it may appear, may be quite important for us to know, and that's part of the public education that we're beginning to launch, that one should be careful with how they use their head, like not using as a ramming rod as in football and some other sports.
Thank you.
Thank you. Joanne, you want to ask a question?
Yes, I have a question.
Joanne.
Yes. I recently saw a movie that's been out a few years called Under Our Skin on Lyme disease or chronic Lyme disease. And a researcher said that when he looked at 10 brains from the Alzheimer's brain bank, 7 out of the 10 had the Lyme spirochete. Is there any association that you know of with Lyme disease and Alzheimer's?
No, I don't, but that's a relatively small sample. I don't know what this observation was, how generalizable it is, but it's certainly something worth following.
And that's why longitudinal studies of just all these features that Dr. Benson, pointed out by his keen observation in 1988, and Bruce Miller at UCSF did a similar thing with something called behavioral variant frontotemporal dementia. And now Dr. Miller is like looking at these patients with behavioral dementias that also can happen very early. And he's seeing some midlife psychiatric diseases, right? And if you don't have this observation, if you don't look at a lot of people, you're in the dark. You're just in the dark. So, yeah, I think this is a very important population.
These longitudinal studies of patients over the course of many years is incredibly important. Some of the most important insights in medicine have come from that. We've learned quite a bit from the Framingham study with respect to the longitudinal study of vascular diseases. We were hoping to set up something similar to that with aging populations longitudinally, create a national cohort. But that hasn't received much attention or enthusiasm because scientists want to worry about getting their next grant, and having such a large cohort is not very sexy. Not too many people want to put their energy into that. They would rather do a small sample study, a narrow question over a defined period to get their next grant. So that's an ongoing effort trying to create an international longitudinal study as a resource for these kinds of questions, so that we could follow people over the long course of their lifetime, with the idea of looking for early lifestyle or early genetic or early markers, how they progress, how they evolve. That kind of a knowledge would allow us to develop the preventative strategies, because ultimately that's where the payoff is going to come. Because by the time we recognize the symptoms of these complex neurological disorders, it's too late. It's already, so much damage has been done that it's very difficult to reverse the process. So the trick is to detect it early. And that has been a difficult challenge to launch. As I said, the scientific community does not see an immediate payoff in that kind of effort. So it has to come from policymakers, and policymakers usually don't take the long view.
Of course. Thank you. It's been a great talk, Dr. Khachaturian. Thank you so much. I have to take one last question if anybody has one. Yes, Dave? Go ahead.
You know, one of the things we talked about in the past is genetics, and so far no things have been found that relate as far as PCA and genetics and family, but I'm wondering if things like Shirley's father had Alzheimer's, if PCA patients, maybe the family members back have Alzheimer's, but maybe not PCA. And I'm wondering if we need to start looking at some of those aspects of the genetic part of it, maybe not just PCA, but things like Alzheimer's versus PCA.
I think you make a good point. I would look for that kind of family history, whether there was a history of not only Alzheimer's, but other comorbid conditions, such as whether there's a history of vascular disease or some other condition that could be the triggering point. It's very likely, this is a speculation on my part, that you have a cluster of genes that could set the stage for the brain to be vulnerable to degeneration, but then the expression of whether, as the disease starts, the degenerative process starts, another factor could influence which direction the degeneration is going to take. And that's what determines whether it's going to be PCA or whether it's going to be Lewy body or whether it's going to be Alzheimer's. It's like a tree. The branches, you know, you have a basic trunk that's common, but then you have the different branches branching out different ways. What determines that from one to the other, we don't know, and I don't have a foggiest idea. But that's the kind of model that I would be looking for.
Perfect. Well, thank you. This has been really informative. I'm going to post this talk online, Dr. Khachaturian, so I will send you a link. Thank you, everybody. You were great, and I'll see you at our next support group. Thank you, Dr. Khachaturian. Have a great day.
Thank you. Thank you for having me. Bye.